Hi everyone, welcome to Vira Education, and today we are going to discuss a very important microbiological technique for your competitive examination as well as for your BSc and Masters in Life Sciences courses, right? So this technique is very basic, and at the same time this is very important technique for your basic concepts and basic topics, right? So it's a microbiological technique firstly, right? And the technique is gram staining, right? Gram staining. So in this video we'll be covering gram staining technique, right? Its principle, its interpretation, its procedure, right? So these are the things we are going to cover under this topic, gram staining. Okay, so welcome to the class, hope you are doing well. Now let's get started with the class, right? Okay, so first here we have the introduction of this technique, gram staining, right? So gram staining में what we have to firstly understand is this is a fundamental lab technique. Firstly, right? Mainly this technique we cover under microbiology, right? Where we study microorganisms, right? And this technique is mainly used to classify bacteria, right? By bacteria, microorganism, right? So this is used to classify bacteria into two classes, right? Number one, gram positive, number two, gram negative, right? So here we have two types of bacteria. We all know gram positive bacteria and gram negative bacteria. Now, if we need to classify these two types of bacteria, gram positive from gram negative bacteria, then how do we classify, right? So here we have a technique. That technique is gram staining, right? So with the help of gram staining technique, we can classify bacteria into two classes, gram positive, gram negative, right? This much you have understood, right? Now this technique, if we study, right, is discovery. First time this technique was discovered by Danish bacteriologist Hans Christian Gram in 1884, right? So this technique is given name as gram staining, right, after the scientist who has discovered this technique, right, first time. So Hans Christian Gram first discovered this technique, gram staining. So we say, okay, this technique is gram staining technique. Okay, right? So gram positive, gram negative bacteria will identify by using this technique, gram staining, right? And how will classify bacteria into gram positive, gram negative, this we have to understand in this particular video, right? Okay, so let's first study what are the materials, right? What are the agents which we use when we perform gram staining technique, right? So these are the materials we generally use while we perform this particular technique in our labs, right? So dyes and agents, right? So the first dye, here we use two types of dye. Number one, primary dye. Number two, secondary dye, right? Primary dye is crystal violet, right? So in this, whenever you study gram staining technique, there you have to remember names of these two techniques, primary staining and the secondary staining dye, right? So here we have two dyes, primary stain, right? Okay, you can use stain also, dye and stain, right? So primary stain and primary dye, right? Stain, dye, both means same. So primary stain is crystal violet in this case, right? And secondary, right? So this secondary dye, or you can say counter stain, okay, or counter dye. So this counter stain is, here this counter stain is red colored and the name is saffronin, right? Its name is saffronin. So the primary stain is crystal violet, right? It gives violet or the purple color, right? And the counter stain is saffronin and it gives red color to the bacteria, right? Reddish pink color, right? Okay, now first just remember the name of these dyes and agents, right? So the second is mordant. Mordant is a kind of fixture, right? Okay, so here we have fixer or mordant is iodine, right? Fixative agent you can say. Then we have decolorizing agent. Decolorizing agent here we have is alcohol or acetone, right? Alcohol or acetone, right? Decolorizing agent is something which will remove the primary stain, right, which will remove the dye, right? So that particular agent is known as decolorizing agent. Now why do we need decolorizing agent in this technique, staining technique, right? So this we'll understand while we'll discuss this procedure of the gram staining technique in detail, right? Okay, So first you have to remember name of these components. Number one, primary stain. Primary stain is crystal violet, which is purple in color, right? It will give purple color to the bacteria upon staining, right? The second is mordant. Mordant is iodine. It is also known as fixative agent. Then we have to decolorize the first stain, and so for this purpose we use decolorizing agent. And this decolorizing agent is, it can be alcohol or we can use acetone also, right? And the counter stain here we have is saffronin, or saffronin is, what is the color of saffronin? Red. Red is pink. Okay, right. So these are the materials we use for gram staining technique in the lab, right? Now the most important thing here we have is, right, so this is the main basic, right, you can also say the basic concept of gram staining, right? And many a time in viva, right, in viva and many competitive exam or your theory exam in at the time of masters or bachelors, this question many a time you will find this question many a time, right, what is the principle of gram staining, right? If you are performing any technique, right, so principle is very very important, right? So whenever you study any technique, just try to study principle first, right? So here we have the principle of gram staining. What is the principle of gram staining? Gram staining mainly works on the on this principle of 2 points, right? Gram positive bacteria and gram negative bacteria, right? So this thing we understood, right? What is the difference between gram positive bacteria and gram negative bacteria? Okay, if you know, very good, right? If you do not know, right, if you do not know the difference between gram positive bacteria and gram negative bacteria, do not worry. Here we have a diagram to make you understand the difference between gram positive bacteria and gram negative bacteria, right? Because whenever we study this principle of gram staining, it is very essential, right? This is very compulsory that okay, we should know the difference between gram positive bacteria and gram negative bacteria. Only then will we able to understand the principle behind the staining technique, right? So here we have the differences between gram positive gram negative bacteria here on the left side, and on the right side here we have gram positive bacteria. In gram negative bacteria, if you can see here, right, first here we have the outer membrane, right? See, first here we have the outer membrane. See, then peptidoglycan layer in between outer membrane and inner membrane, right? So this inner membrane is plasma membrane. We all know, right? Each and every cell is surrounded by plasma membrane. Then after plasma membrane, we have a cell wall, right? We have a cell wall. Then this cell wall is surrounded by again a external layer, right? An outer layer in case of gram negative bacteria, right? So in gram negative bacteria, if I ask you how many covering we have in case of gram negative bacteria, so you will answer we have here three coverings, right? Am I right or wrong? So here we have three coverings, right? On the right side, gram positive bacteria. In gram positive bacteria, if you can locate how many coverings do we have here, right? So here we have only two coverings. Number one, inner membrane, that is plasma membrane. Then the outer cell wall, right? And that cell wall is peptidoglycan layer, right? Now, the layer here we see which layer is absent here, right? So you will answer the outermost layer, right? In case of gram negative bacteria, we have an extra layer which surrounds cell wall, right? But in case of gram positive bacteria, there is no outer layer, right? So in gram positive bacteria, here we have only two coverings, while in case of gram negative bacteria, we have three coverings. So this is the difference between gram positive bacteria and gram negative bacteria while we cover the cell envelope of these two bacteria, right? Now, on the basis of this difference, right? So we have used this difference of cell envelope between these two types of bacteria to classify them, right? And gram staining method, right? Gram staining method is the method which utilizes this difference in cell envelope to identify, to differentiate between these two bacteria, gram positive bacteria and gram negative bacteria, right? So this is the basic, right? Now you have understood what is gram positive bacteria, what is gram negative bacteria, right? Their main difference in cell envelope, right? Then you have understood the basic components, the basic material dyes we need for gram staining, right? Okay, primary stain, fixative, decolorizing agent, and what else? Counter stain, right? So this much you know. Now it would be very easier for you to understand principle of gram staining, right? So believe me, this is very simple. Gram staining principle is very simple, right? Okay, right. See, first you have to write that in case of gram positive bacteria we have a thick peptidoglycan layer, right? In case of gram positive bacteria we have only two layers, right? Outer layer is absent, so this peptidoglycan layer, this layer is thick, right? In case of gram positive bacteria, this cell wall peptidoglycan layer is very thick, right? So you have to remember this thing. While in case of gram negative bacteria we have a thin peptidoglycan layer. So the cell wall of gram negative bacteria is thin and cell wall of gram positive bacteria is thick, right? So this thick and thin you have to remember, right? Now this layer traps the crystal violet stain. Now see, one layer is thick, right? In case of gram positive bacteria, this layer cell wall is thick, and in case of gram negative bacteria, this peptidoglycan layer or cell wall, this is thin. Now just tell me, right, which out of these two membrane, which membrane will bind to dye more efficiently, right? Which will trap more dye, right? This thick one or thin one, out of these two which membrane trap more dye, right? You will say okay, the layer which is thickest, that particular layer will trap more dye, right? Compared to the thinnest layer, right? Now see, so this thicker layer will trap more dye, right? So I'm writing down here that this will trap more dye and the thinner one will trap lesser dye, right? So here we have this principle, right? So when we are going to apply primary dye, that is crystal violet, on the gram positive bacteria, right? And in gram positive bacteria, now we know that this bacteria has this thicker layer, right? So this thick layer will hold or trap dye, right, in large amount, right? And in case of gram negative bacteria, this layer is very thin, right? So it is going to trap less dye, right? So this is the difference between gram positive bacteria and gram negative bacteria at the first step, right? At the first step, while we apply gram stain, right? The first stain, the primary stain, crystal violet. Now just answer me, right? Okay, here we have one slide and here we have second slide. On the both slide we have applied we have applied primary stain, right? Primary stain is crystal violet, right? So let's say here I have gram positive bacteria and here I have gram negative bacteria, right? And on both slide I have applied crystal violet dye, right? Now see, this crystal violet dye will give color on this plate as well and on this plate as well, right? On both the slides this crystal violet will stain the bacteria, right? Now see, in case of this gram positive bacteria, this dye will be trapped more efficiently, while in case of gram negative bacteria, this dye is trapped less efficiently, right? So this is the difference, but both the bacteria will uptake this stain, right? Both the bacteria are stained by crystal violet at first step, right? So tell me, right? Tell me that at this step, right, what color will we have? Right, so at this step both the bacteria, no matter gram positive bacteria or gram negative bacteria, the color will be purple color, right? The violet color, right? So this is how they will ask you in exam, right? Crystal violet will stain which bacteria, gram positive bacteria or gram negative bacteria? You have to answer that okay, this crystal violet will stain both of the bacteria, gram positive bacteria as well as gram negative bacteria, right? And in principle you have to mention that okay, this gram positive bacteria has thick peptidoglycan layer, so this layer traps the crystal violet stain more efficiently, making them appear purple after the staining process, right? While this gram negative bacteria have a thinner peptidoglycan layer and an outer membrane containing lipopolysaccharide, right? So initially तो क्या होगा, gram negative bacteria will take this dye crystal violet and it appears purple blue in color, right? Now after a while, right, after a while at the step number 3, right, at the step number 3 we will add decolorizing agent, right? Decolorizing agent will decolorize this bacteria, right? It will try to decolorize, it will try to wash the primary stain, and primary stain here we have is crystal violet, right? So this decolorizing agent, right, decolorizing agent is mainly acetone or alcohol. So when we wash slide with alcohol or acetone, we'll see a change in color, right, a change in color. See, here I have gram positive, here I have gram negative. After crystal violet, I'll apply decolorizing agent, right? Decolorizing agent, alcohol or acetone will wash these slides. Now, after washing, what will we see that in gram positive bacteria, the color remains same, right? The color remains same. It remains purple color, right? It remains purple or blue color. While in case of gram negative bacteria, this slide is completely washed, right? So it will, now tell me, right, this slide will have color now or not? It won't have any color, right? Because this membrane of gram negative bacteria is not so efficient to hold or trap the crystal violet dye because of its thinner layer, right, because of its thinner cell wall peptidoglycan layer. So this won't be so much efficient that it can hold crystal violet, right, at the decolorizing step, right? While in case of gram positive bacteria, the layer is thicker, it keeps its dye as such, right? So this is the difference. So after crystal violet stain is washed during the decolorization step, and then we add another dye, counter stain dye, that is saffronin dye, right? Now this gram negative bacteria will take saffronin dye and it will appear pink in color, while this gram positive bacteria remains as such, right, in purple color. So at the finally, right, finally after the experiment, when we observe our these two slides under the this microscope, what results do we get, right, what interpretations do we get? We will observe that okay, this gram positive bacteria will look purple in color and gram negative bacteria will appear as pink in color, right? So this is the interpretations, final interpretations of gram staining, right? Now in principle you have to write all these things, right, about crystal violet. Then in gram negative bacteria you have to mention after crystal violet decolorizing step, then after decolorizing step when we add counter stain it will appear pink in color. You have to mention these colors also. In case of gram positive bacteria, purple or bluish color, and in case of gram negative bacteria, reddish pink color, right? So this is the principle of gram staining. Okay, now see, next we have the procedure, right? Next we have the procedure of gram staining. Now if you see the procedure, see, number one, we apply crystal violet, right? Crystal violet application, right? So here we have a slide, right? Here we have a slide. Okay, let's say here we have a slide, right? So this is, okay, slide here I have drawn, let's say this is one slide, right? In this slide, here I have shown you bacteria, right? So these are the bacteria. Okay, these are the different types of bacteria, right? Now we have to identify, right, out of these bacteria, the gram negative bacteria and the gram positive bacteria, right? Now first step is crystal violet application. This is the primary stain and this dye is basic dye, right? Crystal violet is basic dye. Now at the first step we will add crystal violet on the slide, right? After adding bacteria on the slide, we'll add crystal violet dye, and crystal violet dye, after staining with crystal violet dye, what will we see, that bacteria will uptake this color and appear purple or bluish in color, right? Next is fixation, right? We'll fix this dye, right, with the help of iodine, right? Then after this, number three, decolorization step, right? At decolorization step we'll add alcohol or acetone to wash the slide, right? And after washing the slide, what will we see? We'll see that some of the bacteria retain dye, right, these one, right, these one, and the other type of bacteria, right, some other type of the bacteria, few bacteria will wash their color, will wash out their color, right? So this is the difference we see. So here the decolorized bacteria, right? The decolorized bacteria are the bacteria, those are gram negative bacteria, right? Because they have thin cell wall and that cell wall is not capable of retaining the first dye, crystal violet dye. While on the other side, these colored bacteria, these are gram positive bacteria because they retained their dye as such, right? Now in the next step we have to stain again the gram negative bacteria, right, to visualize under the microscope. See, only after staining will be able to visualize our slide under microscope, right. Now, so see, here we can only visualize gram positive bacteria. Now we have to visualize gram negative bacteria also. So for that we have to do counter staining, right. In counter staining, what will we see? See, gram positive bacteria have their dye as such, right, this crystal violet, right, so they are purple in color already. Now the other type of bacteria, the gram negative bacteria, those bacteria, right, those colorless bacteria, right, because the dye has been washed on in the previous step, in the step number 3, when we washed with the help of alcohol or acetone, their color has been washed away, right. So these gram negative bacteria, on applying this saffron in stain, will uptake this color, right, will uptake this stain, saffronin, and as a result, right, after staining with saffronin, gram negative will appear as pinkish in color, right. So this is how we perform gram staining. Now after gram staining, right, once we have followed all of these 4 steps, right, crystal violet fixation, that is iodine, decolorizing alcohol, and then counter staining by saffronin, right. Now we have to see our slide under the microscope. Okay, now when we visualize, when we interpret our slide with the help of microscope, what we see, right, what we see in microscope is that gram positive bacteria will appear purple in color, right, and they retain the crystal violet stain, right, because the crystal violet stain is the stain which is purple in color, right. So we are saying that gram positive bacteria retain their primary dye as such, right, while gram negative bacteria will appear pink in color, right, will pink appear pink in color under the microscope, right, and the pink dye is the counter stain dye, that counter stain dye is saffronin, right. So gram negative bacteria are stained by saffronin, and gram positive bacteria are stained by crystal violet dye, right. So out of these two bacteria, which bacteria retains crystal violet? You will answer gram positive bacteria, right, and gram negative bacteria are washed away, right. So the final results are like this, right. So this is the picture of your microscope, right. After visualizing slides on microscope, we find these 2 types of results, right. On the left side we have the bacteria which is pink in color, right. So this pinkish bacteria is the gram negative bacteria after counter staining with saffronin, right. And this right side, this is gram positive bacteria, and this is purple in color, or the blue is purple in color, right. And this retains primary stain crystal violet as such even after washing, right. So this is how we perform gram staining, right. And this is the principle of gram staining, procedure of gram staining, the basic introduction, right. What is the utility, right, what is the utility of gram staining? Gram staining is used to identify gram positive bacteria and gram negative bacteria, right. And it has many application in your clinical testing, right, in hospitals. Okay, one more thing, right, our E. coli bacteria, the most common bacteria is gram negative bacteria, right. So this is the topic for today's class, and so with this we have completed this technique, gram staining technique, right. So thank you, all the best.